And please keep in mind this order. We have a cells is the basic unit, and then the same cells with the same function together they will make a tissue, and the tissue is make different kinds of tissue with different function together they make an organ. And some organs in our bodies will um collaborate to form an organ system, and organs and some organ system will make an organism, like we have in the example here in the pigs. The lung cells are with the same function, the same structure, it make the same makes the tissue, varied tissue make the lungs, and the lungs is a part of respiratory system, and together with other organs or other or organ system, like so respiratories with the circulatory system, digestive system, reproductive system, we make a whole bodies. So the questions might appear in our final is could be like this: which description of the heart is correct?
I think B.
Sorry, it's B or okay, just only one student. So for the other, or feel free to send me the answers so I can evaluate yeah, the level of student. And also this teacher class is a chance for you to make something wrong, okay? So and then I hope that in your final exam you will increase the possibility to make the right answer, okay? So feel free to send me the answers. Feel free to send me what your thinkings, and then I will know that um what is the level as my student. So here the B or this correct answer, I agree. So the heart is an organ, and this organ contains several type tissue, and they are a part of circulatory system. So I can change the subjects here, or I can change the order, the answer. So I have a new question to put it in your final exams, okay? So another question. The diagram shows a structure found in the human body. Can you tell me what is this?
I think the answer is A. Is A.
So one answers. Yes, this is eyes. So one answer, and only one student send me the answer. What about the other? You don't understand, or you don't want to send the answer, or you cannot give this answer. This is just a multiple choice question. Please express what you think. So okay, this is an eye, right? So an eye is just only an organ. Alright, so this order we made here is only for multicellular organism. But besides, the living organism on Earth can classify into two types, unicellular and multicellular. For the unicellular organism, it can made of prokaryotic cell or eukaryotic cells, but for the multicellular, only made of eukaryotic cells. No reverse case. Okay. We cannot find any multicellular organism made of prokaryotic cells. And the next part of this lesson, we study about the modern cell theories and the old ones proposed by Sladen and Swan. So I don't require you to remember anything or everything, even though a student who want to achieve or the good score, or I just want you to remember to keep in mind for me about what one highlight point, the difference between the modern and the old cell theory. It is spontaneous generation do not occur, but in the past, they do believe that the cell formed by free cell formation, similar to the formation of the crystal. It means that they believe that if we put everything inside flask, we put water, we put vitamin, macromolecules, and provide suitable temperatures, atmosphere, we will have the cells. But snails for the modern cell theories disagree with this. So spontaneous generation do not occur, and all cells come from the pre-existing cell by division only. So in the second part of this, in the second part of the lesson 1, we study about the domain of life. So can you tell me how many domains do we have and how many kingdoms? So all the living organism on Earth can divide in how many domains?
3 domain and 6 kingdoms.
Oh. Yes, here we have 3 domains, and in total we have 6 kingdoms. So here we have 3 domain. The first is eukaryote, second domain bacteria, and third domain archaea. And in some book they merge these 2 domains into the domain Monera. But no, we study 3 domains. And in the domain eukaryotes, we divide it into 4 kingdoms: plant, animalella, fungi, and protista. So in total, we have 3 domains, 6 kingdoms. And the cell on Earth may divide into 2 types of cells: eukaryotic cell and prokaryotic cells. So 2 types of cells are 6 kingdom, 3 domains. And so that's the domain is more is larger and the more inclusive category than the kingdom. So they are bigger, too easy to understand. And what else? So okay, so in this part, in my slide, I have a slide that I captured from the video that they compared their comparison between the eukaryotic cell and prokaryotic cells. What are common between them, what different between them. So you need to rewind this slide again. This is important slides, okay? So we move to the question. So the root hair cell found on the plant root. So which feature could be present in the root hair cell but not a neuron cells? A&D, nothing is A&D. So I just heard 3 answer from only 1 student who is speaking a lot and who speak a lot, and only 2 other students send me the result. What about the other? Trying to give the answer by your own, and this is the easy one, okay? This is for a chance for you to practice. before the final exams. So here's, or I asked about the plant cells, and here the neuron cell is the animal cell. So I just require students find down the difference between the plant and animal cells, and the difference between them is the cell wall. The plant do have the cell wall, but not the animal cells. So in the previous class, when I saw here the sperm cells, student makes confused because they said that the only eggs have a more mitochondria, but not the sperm. And when I put here the sperm cell, they just wonder about the mitochondria answers. But now actually we do have or just a little, just a few mitochondria in the sperm cell, but compared to the number of mitochondria in the egg cell, we can say that we receive all the mitochondria from our mothers, not from our father. Okay. So now I change it to neuron cell to this question is more clear. So B, the correct answers. We only found the cell wall and the plant cell, but not the animal cells. Next question.
I think the answer is B.
So the students who talk a lot, so please send me the answer from the chat box. Okay. So I'm afraid that if you give the answers too soon, the other student is too lazy to think. Okay. Okay, more answers now. All right, good job. It's the time for you to practice something. Okay. I see some always the same like other class. Some choose some choose B and some choose D's, but more of students choose B's, and B is the correct answers. Sarah, a student. Here, for a plant cell do have the cell wall and the shape of the plant cell. Most of the case, they look are like in the shape of the rectangle. So here's plant cells, and this is the hairy root. So they have a part of the apical meristem. So it's very long here, and one or so one characteristic that it's easy to recognize or some cell is or plant cells. That's here you can just see they have or the plant cell have a or a very huge central vacuole. Only plant cell have the central vacuole, but not the other. So for the D's, is somehow look like a slide of the stem of the plant, right? You think that's the plant because it's look like a slide stem. Các bạn nghĩ là có rất nhiều bạn nghĩ là cái phần D này là cái tế bào thực vật đúng không? Mình nhìn nó hao hao cái lát cắt của cái thân đúng không? Kể cả đây là cái lát cắt của thân thì thực ra câu hỏi về cell cô chẳng bao giờ đưa một cái lát cắt thân thực vật vào đây cả. So or B, the plant cell is actually the shape of the hairy root, các rễ tơ thực vật ạ. And A is the neuron cells. C's look like sperm cells, and these are I'm not pretty sure, but it might be the red blood cell. And our next question, which the following may have the cell wall? So this question is more advanced. Now I classify them as more than 15 in 15 point. Because you see, instead of asking which kingdom, the cell from which kingdom have the software, I put the candidates. I put the Unix algorithm here, and you need to classify by yourselves. This algorithm is belong to which kingdom, and this kingdom, or do the algorithm in this kingdom have a cell word or not? So, or some A, some C. Okay, some D also, and we have a B also. Alright, so the Highgate joint is A. Maybe A. Okay, so we go step by step. Okay, so we classify this algorithm by our own. So first, A. What is equalize? Equalize, I think I mentioned a lot of equalize in my lecture because they are really famous. You can find numbers of equalize in your digestive system, right? In our bodies, we have numbers of equalize in our body. So for equalize, they are bacteria, and in bacteria, they do have a cell word. So first candidates do have the cell word. The yeast are yeast cell. What is the yeast? The man, đúng không ạ? So the man, the yeast cell is belong to the fungi kingdom. And for avocado is one kind of fruits, right? Quả. So the cell for avocado is the plant cell, and they do have a cell word. Hairy root. This one is the hairy root, or the shape of the hairy root here. Zeta form the plant root, so they are plant cell, and they do have a cell word. So A, B, or I think this is the best candidate. So we check other candidates together now, right? Are yeast cells? Yes. Colon cells. This is one kind of cell in our digestive system. Streptococcus pneumoniae. This is one kind of bacteria live in our throat, our sore throat. And C, so colon cell is not here, so cannot be. C, we have the X. No X. X is animal cells. Yeast cell, okay. Algae, some of algae, most of the algae is plants, and some of algae are prokaryotic cell. They can be cyanobacteria, so they do have the cell word. Amoeba. Do you know what's the amoeba and the Vietnamese name of amoeba? You know, that's most of the video as I showed you. This I took from the amoeba sister channel. No, so what is amoeba in Vietnamese? So here, amoeba là trùng amoeba. Các bạn nhớ con này không ạ? Trùng amoeba hay là trùng biến hình? Amoeba. So this amoeba is belong to protista, giới động vật nguyên sinh. So they don't have a cell word. So D, amoebas, no. E. coli, yes. Yeast cell, yes. So in total, we have A is the correct answer. And you see, we have six kingdom, right? So only animal and the protista. These two kingdom, the cell belong to these two kingdom, don't have the cell word. And for the rest, they do have a cell word. In the final, maybe I will change. For here for something. So how can I change? Uh, Here equalize famous are bacterias. Maybe I will change into another famous one, like Clostridium botulinum. Itself a change into the mode or somehow like this, right? Từ nấm men chuyển thành nấm mốc ạ. And pay attention for me. I give you a hint here. So any species on Earth do have the Latin name. This is the Latin name. And whenever you see the Latin names, it means that the organisms is belong to only fungi and bacteria. I will not put the Latin names of the plant and animal here, so it's make you confused. So Latin names, bacteria or fungi, okay, in your final exams. Our next question is easy one. So most of the answer is A. So one answer is D. For the student who drew this, I think you need to read the questions more carefully. The question 1 to ask which belong to prokaryote but not eukaryotic cells. And the difference between them is that eukaryotes, eu is mean two, and carry here two nuclear. So eukaryotic cell do have the nucleus. Here, this belong to eukaryotic cell. Nucleolaminar is one part of the our nucleus. The underneath this our nuclear membranes, we have nucleolamina, and also nuclear envelope here is belong to nucleus also. So BCDs belong to nucleus, not prokaryotic cell. Prokaryotic cell, pro is mean before. Before it meant carry is nucleus. So before nucleus. So Vietnamese we have sinh vật nhân sơ hay sinh vật tiền nhân ạ? Tiền đúng không? Before tiền nhân ạ, sinh vật trước khi mà nó có nhân thực. And for the prokaryotic cell, we don't have any nucleus. They do have one reason containing genetic material we call nucleoid, vùng nhân ạ. Nhân sơ chỉ có vùng nhân thôi, còn nhân thực thì chắc chắn là có nucleus rồi. So it's related to the slide that I mentioned before. The slides or the comparison or the characteristic of the prokaryotic and eukaryotic cells. This is important. And now we move to the lesson number 2. So you know that's when we're looking for a living organism, looking for some characteristic like digestion, respiration, reproduction, excretion. So it means that's when you within the cell, only single cell can express these kinds of characteristics. So that's why we call them the basic units of life. Only single cell can express all the characteristics of the living organism. And it's just because inside a cell they do have structure, they do have the organelles to mix all these characteristic that I mentioned: digestion, respiration, reproduction, etc. And now we study about the or cell structure of actually the eukaryotic cell. So in the eukaryotic cell, we have a mitochondria, we have a Golgi bodies, we have a ribosome, we have a lysosome, cell membrane, nucleus, endoplasmic reticulum, cytoskeleton, and also the cytoplasm. All right, so the question may appear as like, where the site the protein synthesis in the cell? So, so, okay, sorry, here, for the endoplasmic reticulum, we have two types of endoplasmic reticulum. We have rough ER with the ribosome stick on it, and we have smooth ER here. So ER, rough ER, and smooth ER. Okay, so come back to the question. I received one answer, two answer. Which is one, three. Our mode is 2D. A figure to C, 1 to 8. So here, the sign, the protein synthesis in the cell we have, they are chromosomes. Chromosome is the sign where the translation happen. So when that the ribosome is stick on the rough ER, so if the ER appear, it could be the rough ER, not the smooth ER. There's no ribosome in the smooth ER. But even if I put it here, one more, one additional answer, like rough ER here, the answer is still ribosome, because ribosome is the side protein synthesis. Even in the rough ER, the side protein synthesis still the ribosome stick in it. Okay, so this question, ribosomes. Next question. The sign with high level of hydrolytic enzyme activities. What is this? Yes, this is a lysosome. And that in for example in the macrophage is our humoral cells. The macrophage go to engulf or these bacteria attacks our bodies, right? So when the bacteria see some strings or not, when the macrophage see some strings, meet some stringed bacteria, they will engulf, and with the help of the lysosome inside the macrophage, they will digest these kinds of bacteria. So the lysosome here is working like our stomachs have to digesting. Our next question, the sign which generates most of the chemical energies. So this is the mitochondria, and they are the energy factory in the cell, right? Energy factory in the cell is the adenine. So they generate the energy in the cell in the form of ATP, adenosine triphosphate. We have ATPs. So it's also called the energy monies inside cells. So this is the mitochondria, and the next the sign with steroid hormone synthesized from cholesterol. Thus, we have smooth ER. The sign of detoxification of drug and harmful chemical. Smooth ER here. So what is the sign of detoxification of drug and harmful chemical? Are not mitochondria, not bacteria. Oh yes, smooth ER. So both of these functions is the function of the smooth ER. So you know the detoxification are here detoxification organs. In our bodies we have the liver. So when you eat some things or you drink some things, like when you drink alcohol, the ethanol will be absorbed from our intestine. The ethanol go with the bloodstream to go the first part in the liver. And in the liver, and also the liver cells, they in trust of detoxifying the ethanol, they transform the ethanol becomes or acetaldehyde. One compound is less toxic, and so on, so on. So many reaction happens to detoxifying the ethanol in our bodies. So the sign detoxification of drug and harmful chemicals, we have smooth ER. The sign are steroid hormone synthesized from cholesterol, we have a smooth ER. But the sign in trust of produce the proteins, especially protein for export, we have the rough ER. And the site of protein synthesis is the ribosome on this rough ER. I hear for the ribosome here we have two kinds of ribosomes. One is ribosome is in the rough ER, and one ribosome is free floating in the cytoplasm. So the ribosome in the rough ER is most their function is produce the proteins, but mostly for the export, for secretion. But free floating or ribosome is most of the case they produce the protein which is used inside the cell. Next question. The organelle which has ability to deliver vesicle, a package of various cell products. Yes, they are Ganji bodies. So here's now the function of the Ganji. Next question. So in the previous question, it's just basic information, basic knowledge. And so this is an advanced question where you need to combine the function of all the organelles here to make the correct answer. So in this question, I classify them as more than 16 with 20 points. So advanced question for the students. One D's, one B, and what else? A's. Oh, C. So now we have enough ABCD. Okay, we go step by step to solve this question. Okay, so we can see how the organelle inside the cells are working together. Here, this is production of an enzyme for secretion. This means that we want to export some enzyme, and enzyme is more of the case they made from the proteins. So when we want to extract the enzymes, or we want to produce enzymes, it means that we started from the gene coding for this enzyme, this protein. So here the genes are coding for the protein. So if we want to produce something, we started from the brain of the cell, which started from the nucleus. Nucleus will make the command or give the or make an order to send to the cytoplasm. So here nucleus. Nucleus, and they send the command, they send the order in the form of mRNA. mRNA messenger RNA, they send an email to the cytoplasm. So in the first destination of this email, they will reach the site protein synthesis ribosome. Ribosome here in the rough ER, remember, rough ER is just because secretion. We want to export something, so is go to meet the ribosome in the rough ER. So now we have the translation process, protein synthesis. So here proteins appear. But what happens with these proteins? It is mature ones. No, this protein is just simple, like a chain of amino acids only. So this is not mature ones. And the next we or if they follows after rough ER, they will go to smooth ER. And from the ER, if you want to send to the Golgi bodies, it's packaged in the form of the vesicle. And they send this vesicle to the Golgi bodies. Do you know which phase of the Golgi body we receive? Cis phase or the trans phase of the Golgi body we receive the vesicle. So I told you before the seed. Seed is mean or a similar sound like receive, right? So they send first in the seed phase, and after that the Golgi will transfer molecule through the trans phase. Remember? So here in Golgi bodies, they will packaging, they will modify, they will add in some elements. Or so in Golgi, it's simple that they make the protein become the major one, and this is if it's an enzyme in this step, the enzyme have the activity and they become the active compounds. It's not form. It's not like the protein here, the premature. And after that, it's going to send out and the package in the form the vesicle to send out of the cell. So now, what could be the correct answer? Here, so we started from A. So A, if we start is nucleus, is this okay? So it's quite okay. We started from the brain of the cell, nucleus, to make the command. This command come to our ribosome. The sign the protein is still okay. So it send protein to Golgi apparatus. It's still okay, but no way they come back to the rough ER, right? So it's not okay. B, start with nucleus. Okay, send message to the ribosome on the rough ER. Still okay, but the forming the vesicle is still okay, but it's not a vesicle is no way to come back to the ribosome. C, started with the ribosome is still okay, then now we don't have the nucleus, right? Our send packaging in the form of vesicle still okay, but send it to the rough ER is not okay because the ribosome is stick on the rough ER. We don't need to form the vesicle to send protein from a ribosome to rough ER because they are in the same more or the same area. So we have only D. Start with ribosome, go for rough ER is still okay. No vesicle here is okay. Are sent to the Golgi apparatus, packaging the vesicle, and then sent out. So here D is the correct answer. Congratulations, the student who have the answer is D. So in the final exams, I will change this question a little bit. Like I will change here into an hormone, for example. So hormone, it could be our steroid, or it could be the proteins. If they're steroid, we can start it from instead of rough ER, we can put here smooth ER. Okay, so it depends, and also these sequences I will change, but if keep the right order, it's still the good answer. Okay, I so next part 3 here, part 3. So in the part 3, we study about the chemical component of the cells. We study about different chemical bonds, and we study about the macromolecule, right? And here, this is the graph of atoms. And atoms composed from neutron, electron, protons, etc. So it's chemistry, so you don't need to remember. So, or please give me answer. That's what determines how the atom interact. So remember the outermost shell, the electrons in the outermost shells, if they have incompletely filled electrons in the outermost shells, it make them less stable. So this atom want to give or receive electron to achieve a complete outermost cells. So it's leading to two kinds of chemical bond, covalent or ionic bond. And here, if they have the complete field electron cells, they don't need to give or receive, and they become these inert gas cho nên khí chơi. So, here is the bond strength in biology. I need to emphasize so many times in biology. So we have a covalent bond, the strong bond, and here is the weak bond. It's different in chemistry because in chemistry, ionic bond is the strong bond, and is more or less like covalent bond. But in biology, you know that 70% of our cells are water, and is mean that or in our cell, this is the aqueous environments. So in water environments, the ionic bond is a weak bond. So the bond strength is only occupies in biology, okay? It's not in chemistry anymore. And also, in biology, for examples, in the hydrophobic interior proteins, the ionic bond can even upward the strength of the covalent bond. Yeah. So pay attention, or in so many students choose the ionic is strong bonds, but it's wrong. We study biology now. All right, so question, which bond is the strongest and the most stable? So it's covalent bonds, right? Easy question. But I think it's appears so many times in the exam. Are which bond hold complementary strand of DNA together? Yes, hydrogen bond. Here we have CG by 3 hydrogen bond and A has 2 hydrogen bond with T, right? Next question, which bond are responsive for determining the three-dimensional structure of fold proteins? So in this question, actually they are just three weak bond. Any weak bond can join in this process and to make the three-dimensional structure of fold protein. Next, in the covalent bond, electrons are, and in ionic bond, electrons are. So this question, I think, is appears more than 3 times in my exam. So in covalent bond, electrons are share, and in ionic bond, electrons are gains or loss. Okay. So in the second part of this lesson, we study about the component of the cell by the compound. In organic compound, we study about the water. 70% in our body is water. About the organic compound, we study 4 macromolecules: carbohydrate, lipase, proteins, and nucleic acids. So questions: what is the smallest units of proteins? Oh, most of the case B, amino acids. I agree with you. The smallest unit of proteins is the amino acids. Our still have some have some students made a mistake when they choose C. So so student who have the answer C, you made a mistake because we want to correct the smallest units are amine is one of the 3 main parts of amino acids, but the smallest unit of protein in amino acids is just like we said that the sound is the fundamental basic unit of life, and inside the cell we have a number. We have a various organs or structure, right? So we just say the cells is the basic units, but not the cell organelles. There's someone to these are totally wrong. Sorry for you. And our next question: with this small unit of nucleic acid? So nucleotides, right? Our next question: with the small unit of polysaccharides? Our monosugar or monosaccharides too common. Monosaccharides too common. We have the monosugar. Next, with the smallest of units of fat? So the fatty acids, right? And the name of the reaction that's from 2 monosaccharide form disaccharide. Yes, they are condensation, and the reverse reaction we have a hydrolysis. So, can you tell me the linkage between two amino acids, two nucleotide, two fatty acids? So, A1 connects with AA2, AA3 by weak linkage. And peptide 1, peptide B, peptide 3, peptide 4, weak linkages. So here these ester. Yes, for nucleotide, first for the ester. Okay, and in the lesson, please pay attention for me the for level structure of the proteins. Okay, so here is to sum up this lesson. You can see we have a subunit sugar, amino acids, nucleotide. With covalent bonds, with a strong bond, they will connect these kinds of the subunit together to make a macromolecule. And here, like amino acids, we make the proteins, nucleotide, we make the RNAs. And by the weak bond, the non-covalent interaction, macromolecule can assemble together to make the structure inside cells. Like we have proteins and RNAs make together, they make the ribosome, which is the site of protein synthesis inside cells. Okay, this is conclusion of this our first lesson. Here we have another question. It is easy ones. So I hope that you have the correct answer. All of you have the correct answer. So we have a B, a chain of amino acids. So A, carbon, hydrogens, and oxygen in the ratio 1:1. This means they are carbohydrate. Or for example, D, a series of the nitrous bases attached to a sugar for fat backbone. We have our acid nucleics. Okay. Next question. I think it is an exercise and it's quite familiar with you in high school, right? I think you do it a lot in your high school. And so we have a DNA fragment. We have a total basis, and we have a number of hydrogen bonds. And now you need to calculate for me the number of X bases. And for cytosine here is equal to the X in your high school. So in this question, you need to or propose two or equations. Okay, so in general, a plus t plus g plus c equal to 1,500. But a equal to t and g equal to c, so we have first equation 2a plus 2g by equal to 1,500. And because A complementary T by 2 hydrogen bond, G complementary with C by 3 hydrogen bond, and we have second equation 3A plus 2A plus 3G equal to 2,000. So to solve this equation system of equation, we have A T 250 and G C 500. Okay. And next question is about the DNA also, the structure, DNA structure. So more the answer is N. So M is G, so it's mean, and is C. However, we have, what's here? And G and C. So however, this is the original DNA strand. So we have a G here and then we have C here. And after that, in our DNA replication, we have two new strand form. And here G, C, C, D. So the first G is and the second C is R. So we have two answers, and R are citizens. So congratulations, just only one student have the correct answer. And we move to the next part about parts for the nucleus. In this part, briefly remember for me the river. So remember briefly about the structure of the nucleus. We have a nuclear envelope, so the membrane of nucleus, and stick on it. We have I call them the door between the nucleus and the cytoplasm. They are nuclear pore. And we have a nucleolus, or is where is the sign of the ribosome synthesis. We have also chromatin, is genetic material inside cells. And the question, which type of cell does not have a nucleus? Yes, so easy question. Prokaryotic cells, we have only two cell types, and which cell type do not have the nucleus, it means they are prokaryotic cells. The cytoplasm have a similar function to which part of the nucleus? So we have, so for example, we have cell membranes. So in parallel, we have nuclear membranes. So here we have a cytoplasm, and in parallel, we have nuclear plasm. Next question. The name of the structure which is used to transfer macromolecule between the cytoplasm and nucleus. Yes, in one slide, I have a gram that's to show you that's how the HIV virus can hijack these two sequences of our cells, and or to use these two sequences to bring to transport there's macromolecules between the cytoplasm and nucleus of our cell. Okay, so we have two structure. First we have an NES, and second we have an NLS. And NES, E here is stand for export. Okay, so export. So if you want to bring thing from the cytoplasm to or nucleus to cytoplasm, we need NES. And NLS is a nuclear localization sequence. So if you want to bring thing from the cytoplasm to nucleus, you need NLS. Okay. Next question. Yes, there is now nuclear pores, what I call the door between nucleus and cytoplasm. Our next question, the mRNA TNR. So yeah, this is a question. So they need to go through the nuclear pores or nuclear pore complexes, NPCs. So in the second parts of this lesson, we study about the structure of the chromosomes. So if you want to achieve more than 12 parts, just remember for me briefly structure of this chromosome. But if you want to achieve 10 to 12 parts, you don't need. Here are the questions. Which kinds of histone is not found in the histone atomic core? You know, atom is mean something made from 8 unit. Okay. So this is the S1, the linker histone. the linker histone. And for the 8 units of the ultimate car, we have 2H2A, 2H2B, 2H3, and 2H4. In total, we have the 8 proteins. Next question. In the normal humans, in the normal human beings, number of chromosome is so we have a 46. One student 23. 23 is we have a 46 chromosome or 23 pair. Okay, pair is 23, but in general we have 46. Our chromosomes are made of protein and what other molecules? So they are, it is DNA, single or double strand. So DNA is single strand. And which of the following? No, it's not. Which is considered the basic units of the heredity? So this is the genes. Correct. Good job. Next, a pictures of the person's chromosome is called karyotype. Next question. What are the end pieces of the chromosomes and their functions? Bạn nào comment gửi cho cô cả một list dài ngoằng tiếng Việt đi kia. So we have here, this is the chromosomes, right? In the center, we have a centromere. And in the end point here, we have the telomere. So in general, telomere will protect the chromosomes. And so after X cell division, this telomere will be shortened a little bit. So if we cannot maintain the length of the telomeres after 40 to or 30 to 60 times of cell division, the cell will be pushed into the apoptosis. So please have a look back at my slide about three scientists who discover the structure and the function of the telomeres, and also um, and they received the Nobel Prize. Remember, and also they discover about the telomerase. Telomerase is one kind of enzyme which is active is more than 90% in the cancer cell. So it means that in the cancer cell, they can conserve, they can protect the length of the telomeres after the cell division. So after cell division, telomeres will elongate the length of the telomeres. And that's why the cancer cell is become the immortal cell, because they never died. After how many times of cell division, their telomeres remain the same length, so we never put them into the apoptosis. So this is the end of the part 4. We have some more minute breaks, okay? After that, we can come back to continue the part 5. Membrane. So about the membrane, this is the structure of the membrane, and to start, or here we have a question. This only one question, but from this I can modify and create varied type question. Okay, which organelle have a single membrane, double membrane, and non-membrane bound? We have a three organelle have a double membrane. Remember? Um, this is the easy one. So we started first. Which cell have a double membrane? Which organelle have a double membrane? So the first we just study nucleus, right? Second, we have mitochondria, and last we have chloroplast. So in total, we have three organelles that have double membrane bound. So what about the non-membrane bound? For non-membrane bound, this means that these organelles are more solid structure. So they don't need, or they have no fluid filled, so they don't need to have the membrane to cover the inside, right? Non-membrane bound. What is non-membrane bound? Which organelle, which structure is more solid? So first we have ribosomes. Ribosome is made of proteins and RNAs, right? There is no fluid fields, so they're the first candidates of non-membrane bound. Second candidate we have, which is made of proteins. Cytoskeletons. And the third, we have the cell wall. Okay, three three organelle have a double membrane bound, three have non-membrane bound, and for the rest we have the single membrane bound. Okay, so for example, ribosome here, we actually they are not our organelle, they are just a structure inside cells, but I classify them as an organelle for you to easy to remember. Next question, which component in the membrane distinguish the blood type? So here in the cell membrane here we have some side chains. You can see there are some side chains in the surface of the cell membrane. They are carbohydrate chains, right? And they defined the antigen in the surface of the red blood cell. So for more detail, we have oligosaccharides determine the distinguish the blood type. And remember for me, GAND and our GANAP, okay, two types of antigen in the surface of the red blood cell. So if we have a GANs, which blood type do we have? If we have the GANAP, which blood type? If we have both of them, which blood type do we have? And if without both of them, which blood type do we have? So we move to the next part of the lesson. It's about the transportation between two sides of the cell membrane. We have the passive transport. When we need to bring things from the high concentration to the low concentration. And for this, because we bring things from high to low concentration, we do need provide them any energies. And the second mechanism is active transport. Active transport, we need provide energy for this process because we bring components from low concentration to high concentration. And for this part, remember, or please show me the difference between the simple diffusion and facilitated diffusion. So here, simple diffusion. And what we call here the channel mediated is actually facilitated diffusion. So they do belong to passive transport. They don't require energy to bring things from high concentration to low concentration. But the facilitated diffusion require protein channel, while the simple diffusion is not. Next is about the osmosis. Osmosis is movement of the solvent or water, or movement of solvent, and most of the case is the water. You know that 70% in our body is water. So when we call the solvent in our bodies mean most of the case of water. So the movement of the water across the membranes in response to concentration gradients of our solute, and we call them the osmosis. So here we have the inner hypertonics. Hypertonics is um, hypertonic solution where the concentration inside a cell is lower than the outside. So the water move out of the cells and the cell will be shrink. In hypotonics, the concentration inside your cell is higher than the environment, the external environment. So the water move in the cell become swell up and burst. And last thing, isotonic. The concentration in the external and internal environments is remain the same. So the water move in and move out more or less the same. So the cell keep the same shape. They keep the shape, and is more or less nothing changed with the isotonic solution. So can you give me the difference between the osmosis and the diffusion? So one is for the osmosis movement of the solvent, right? But the diffusion is movement of both of solvent and the solute compound. And also, or revise for me the exocytosis and endocytosis. And question: what are three types of passive transport? So correct. All the answer is D. Okay, so whenever you see the pumps, you see the pumps here is mean you need to provide energies for the pump work, right? Đó là cái bơm đúng không? Mình cần năng lượng. So this they are active transport. And whenever you see exocytosis, endocytosis is mean active transport also. So D if is passive transport. So these are easy questions. And now we have one advanced question for a student who want to achieve more than 16 point. A question about the wet skin toppings. I left you 30 seconds to solve this question, okay? So here about the wet skin toppings. They are made of selective permeable, nghĩa là tính thấm có chọn lọc, right? So here demonstrate an osmosis là quá trình thẩm thấu. So when you put the wet skin treat into the solution after you take it out, the weight is increased. It means that the water move from the solution S into the solution R. So it means that what about the concentration of the solution R and the concentration in solution S? Can you get? So in solution S, the our salt solution is smaller than the salt solution in the solution R. So the water will move from solution S to the solution R, right? So R, I see one student can explain about this. Good job. And we move to the next lesson. It's about the cytoskeleton. It's quite complicated, but to achieve 10 to 20 point, you need to remember for me these things are first, that's three main components to make up a cytoskeleton, and shows me some simple questions like what is the basic subunit that form a microtubular in the proteins? So a tubulin, so tubular and tubulin, the sound are like similar, right? So tubulin make up the microtubular, and we have an alpha and beta tubulin together. They make up the microtubular. Our next microtubular are cut bind, wicks and dams. So they are catanins. One names are is named by the Japanese scientists, right? Catanins mean the sword. And our next question, you need to remember for me also the diameters of these three main components of cytoskeletons. So 825 nanometer and also 8 nanometer from 8 to 7 we have the micro filament, and 8 to 12 we have an intermediate filaments, right? So or if the student want to achieve more than 12 party splits, revise for me more details about the cytoskeletons, like how they can transport the vesicle and macromolecule inside the cell, how they can have the cell divisions, right? So how they form the spin the fiber, etc. And if you want to achieve a 12 part, that's all information you need to remember. For me, it is in this slide only. We move to the next lesson. It's about the cell cycle and regulation. So here in the first part we study about cell division, right? So for cell division, I will show you some pictures, and you need to define the weak state of the mitosis in that picture. So we have a anaphase, and our next picture. So we have B metaphase. So, or please rewind a video about those mitosis. Remember, we have four states P met. P is for prophase, M is metaphase, R A for anaphase, and T for telophase. And also M is stand for meter. So you can see that B in the metaphase, all these are all the chromosomes is lined up in one line. So in the meter of the cell. So B is the meter. M is meter, and A is also stand for away. Here, two groups of the chromosome is started to move far away from each other, and they go toward the opposite pole of the cells. If you see just one cell and all the chromosomes started to form, it means that they are prophase. Or you can see one cell with two groups of the chromosomes in the two opposite poles, mean that telophase. If you see one cell um here is more or less form two daughter cells. Mean we have the cytokinesis. Okay, that's all about the cell division. For achieve 12 points, I just require you remember these kinds of information. In the second part, we study about cell cycle, right? So for cell cycle, here we have three phases. That the M phase is mitosis, G1 is grow one, S phase is the synthesis, and G2 grow two. S is mean in the S phase synthesis, it's DNA replicate in this phase. And between each phase, we have different checkpoint. The checkpoint is like our final exam, right? If you pass the final exam, you will go to the next level. If you cannot pass, you stay there, and sometimes you need to study again this subject. Okay. Um, the question about the cell cycle. The cell may withdraw from the cell cycle at which stage? Now, as you see, I have a slide about this the G0 escape from the end of the Z1 to the Z0. Do you remember which is the Z0 stage? The state of the cell escape or the cell cycle. And in Z0 stage or Z naught or Z nothings, we in Z0, the cell withdraw from the cell cycle and they concentrate or their works. They don't want to divide anymore. So can you give me an example of the Z0 stage of the cell in a Z0 stage? We have neuron cells. Neuron cells is one type of cell that have the or more like the same age like you are. So when you are babies, your brain keep divine, and when they reaches the or I don't remember how million of cells, when they reaches the number of cells, they stop there, and they are withdraw from the cell cycle and go into the Z0 state, and they working from since there is until now. So our neural cell is one famous example of the cells in the Z0 state. Besides, we have some kind of cells in the Z0 state, like some heart cells, some muscle cells. Okay. Next question. Oh, sorry. Stop the cell cycle if DNA damage is detected. So if DNA damage detected, the P53 will stop the cell cycle, right? P53 We stop the cell at the checkpoint and takes more time for the cell repair the DNA damage by calling the DNA polymerase to fix the DNA damage. If the fixation is unsuccessful, the P53 will put the cell into the apoptosis to destroy all the cell with DNA damage. So when the cell have DNA damage, it means the cell have mutation. Tức bị tổn hại ở DNA thì tức là the cells là cái tế bào đó bị đột biến ạ. So we don't want to keep the cell with the mutation, right? But if we cannot put them into the apoptosis, the mutation, the cell will divide, and they will accumulate. They will accumulate, and somehow by sometime they will become the cancer cells. When they are cancer cells, it means they're making a tumor. So the P53 will prevent this happening, and that's why the P53 have the other name is tumor suppressor. Okay, and so we move to the next lesson about the cell communication. About cell communications, we have three stages, like we communicate with each other. We have a reception, we have a transduction, and we have a response. So the first question is about the receptor. So here is the cells, here is the internal receptor, and here is the external receptor in the surface of the cell, internal in the cytoplasm. So they cannot bind to the ligand, do not cross the cell membrane. So here A is not correct. B are protein like G protein Cooper receptor is appear in the external receptor. And C is the correct answer. And the next question is about the ligand. Steroid, a biological active compound, are delivered formed. So cholesterol, right? Cholesterol, no A, sorry. Biological active organic compound is mean they are hormones. Actually, steroid hormones are delivered from cholesterol in smooth ER. Okay. Our next question about the distance of communication, right? The communication between macrophage and telomerase is one example of what? Yes, there are no distant communication. So the communication between two email cells are no distant communication. Like the other way to mention the long distance communication and short distance, no distance, local, and one example in cancer cells. So we have short distance, we have our synaptic. Local. Remember the first slide I showed you about the yeast cell sending the mating factor to the environment surrounding them. So we have the paracrine. No distance, we have contact dependent and example in cancer, of course, outer crine signaling. While the cancer cells secrete the ligand, secrete the growth factors to stimulate themselves. Okay. And here is just more understand like um you communicate with your friend. If it endocrine, this means maybe you make a phone call or you sending an email, right? Paracrine is like you talking in the class. Synaptic or you talk to your friend who's sit next around you. Outer crine is like you talking with yourself. And contact dependent, like you can or only or give them a letter, right? So when you make the contact, you can receive the message. If you're not a contact, you cannot receive any message. So we move the part 9 about the apoptosis, cancer, and the stem cell. Question: this is an active cell death process. So it is hypothesis. Next question. This cannot be killed by hypothesis. So C, cancer cells. Cancer cell, right? We cannot kill them. That's why they become the cancer. Our next question. Oh, sorry, here. The immortality exhibited the cancer cell due to the presence of this question I explained in the slide about the chromosome, right? Telomerase. Sorry. The migration of cancerous cell from the site of origin to other part of the body, forming secondary tumor. And what occur? Still A and D. I think this the framers favorite answer A and D's in all the tutor class I taught this year. So here, when one cancer cell one tumor move from the origin sign to the secondary sign is mean metastasis. Metastasis is quá trình di căn, but angiogenesis is the process where the tumor become bigger and they want to fit the cell inside them, so they sending the masses to the blood vessels surrounding them, so they want the half. They are hungry and please feed me, and the blood vessels surrounding them will move and will develop deep inside the tumor and to provide the nutrition for the tumor. Ở cái câu này thì Cúc cũng hiểu tất cả các lớp mà cô dạy đều có một nửa chọn A và một nửa chọn D, thì không biết là các bạn tại sao các bạn lại bị nhầm ở cái chỗ này nha. Cô nói tiếng Việt một tí nhé, bởi vì thực ra là nó là kiến thức mà thưởng thức mà cô nghĩ là ngay cả các bạn không học ngành sinh cũng nên biết, đúng không ạ? Đầu tiên là ung thư là gì? Đó là một cái tế bào mà nó phân chia không ngừng ở trong cơ thể mình và mình không diệt được nó, thì là mình sẽ bị ung thư và nó tạo nên các khối u, đúng không ạ? Thế thì khi mà một người bệnh mà bị bác sĩ kết luận là ung thư ác tính thì tức là cái tế bào từ cái khối u đó có khả năng di căn, thì đó là ung thư ác tính ạ. Và cái quá trình di căn chính là cái quá trình mà diễn giải trong câu hỏi này, đúng không ạ? Tức là một cái khối u hoặc là một cái tế bào ung thư mà nó có thể di chuyển từ cái vị trí ban đầu của nó đến một cái vị trí thứ cấp. Ví dụ như là một bệnh nhân bị ung thư gan chẳng hạn, và cái tế bào ung thư gan có thể đi khắp cơ thể, đến xương, đến thận, đến các bộ phận khác trong cơ thể, và có thể mọc nên khối u mới ở bộ phận đó. Đó là quá trình di căn. Và một khi di căn thì đúng là cụ đi chân lạnh toát rồi, đúng không? Rất là gần như là không cứu chữa được nữa. Đó là quá trình di căn nhé. Còn cái quá trình angiogenesis đó là một cái quá trình tạo mạch mà thôi, đúng không ạ? Khi mà bệnh nhân có khối u này, khối u lớn lên này, một khi mà khối u nó quá to rồi thì các chất dinh dưỡng không thể nào mà trao đổi với cái tế bào ung thư ở trung tâm cái khối u đó được, đúng không? Thế thì bọn ung thư nó rất khôn, nó sẽ gửi đi một cái message, nó gửi tin nhắn cho các mạch máu ở gần đó là tao đói lắm, đến nuôi tao với. Thế thì các mạch máu nó sẽ phát triển thành rất nhiều các mạch máu bên trong khối u đó và sẽ nuôi khối u. Chính vì thế mà các bệnh nhân ung thư mà có Chị Sạn thì cô không nói nhé, các bệnh nhân ung thư khi mà khối u nó đã to lên rồi thì các bạn thấy hầu hết bệnh nhân ung thư sẽ rất là gầy, bởi vì là ăn được bao nhiêu, người có bao nhiêu chất dinh dưỡng là đi nuôi cái khối u đó hết, đúng chưa ạ? Đây là vừa là kiến thức sinh học mà là kiến thức thưởng thức mà cô nghĩ là bạn nào cũng nên biết. Và câu này là metastasis nhé, không hiểu sao tất cả các lớp mà cô dạy đều chọn một nửa chọn A và một nửa chọn D luôn. So we move to the next question. Now, what does the pluripotent means? So A, pluripotent. So remember, for me also, multipotent are some cell and unipotent for only cell, right? Next question. What's so special about the stem cells? Yes, B, they can mix other type of cell, right? Next question. So for this question, I give you the answer here. The proven of stem cell treatment maybe had some cancers like leukemia or some blood disease or some immune system disease. A, B, C are correct. Okay. We move to the last part about the prokaryotic cell. Here, this is about the cell where prime gram-positive bacteria is true. So all of the above, we have a D. So the cell were comprised of many layers. Actually, he's a peptidoglycan. Here, many layer of peptidoglycans is more precise, right? And it's thicker than the gram negative. It's true, it's okay. So consequently, D is the correct answer. And the cell walls do comprise of the teichoic acid. But I think you don't find you don't pay attention about this information in the slide, okay? All of the above is correct. Next question. This question is, I think it's difficult because it's not appear in my slide. So whenever we see the outer surface of gram negative, you can see the LPS, lipopolysaccharide, is one kind of famous compound that's causing the inflammation in our body. Our next is about the mesosomes. So here is the prokaryotic cells are some the membrane we found so many times to make a mesosomes. And they have to put two DNA genetic material into two opposite poles, and also have of the respiratory respiration process inside the cell, right? So they are more or less similar, like the mitochondria. The mitochondria, you know, the inner membrane, the mitochondria is folded so many times to make a pretty, right? And they have respiration process in mitochondria. So we received the correct answer. Our next is about the viruses. A virus is made of Yes, A, protein coat and nucleic acids. The basic question, right? Our next questions. So, with the following statement are true about the virions. So what is the virions? Virion actually a completed virus. Yes, here we have the answer is D, fully formed. Fully formed in completed virus. It's actually the virus with the capsid protein surrounding with the acid nucleics inside. They are virions. Last question, I think. Last question. So A, C, D, no. The keyword here is or. The nucleic acid, the genome of the virus could be DNA or RNA, but never both of them. So we have the double-strand DNA virus, single-strand DNA virus, or single-strand RNA virus, double-strand RNA virus, but never appear in one virus, both DNA and RNA. So the keyword is or R. So C, the correct answer in this question. So this is the end of the tutor class. Today we have another lesson about the cell culture and application. So please rewind by yourself at home because it's still up here in our final exams. Okay. Do you have any question about this course? Or do you have any question for me? No questions.
Can I ask about the time and the number of the question we will have in the final exam?
I'm not decided yet, but normally it's maybe 45 minutes. Last year it's 45 minutes, I think. Number of question I really don't remember. So because I need to create eight code for the final exam. So it's a lot of question, and I not decided the number of question. Yes.
Okay, thank you.
So if you don't have any questions, we can stop here. So I hope that you will pass my course with the highest score, and I want to meet you as in our new universities, but I don't want to see you again in my retake exam or in my course next years. Okay, so good luck for your final exams.
Thank you,